Okay, children, shall we start then? Want to come? Anrosh. Sinadina. Right. Can you all hear me? Sarah? Wait, Sarah. Sarah, Sarah. 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 Muslim. Okay. You look very happy today. What's going on? There. Everything boy, can you talk, please? I'm happy to hear your voice.
So you have heard my voice. You hear my voice 4 days per week.
Yesterday also I heard your voice.
Yeah, yesterday did I?
Okay.
Satara special guest connecting now.
Moderator,
Ah.
in a guest like thing na done. Guest la good call and the
Yes, sir.
Right. So, why Michelle is silent today? Michelle, is there a problem?
No, sir.
Why? You're having your lunch? Is it?
No.
Yeah, younger sister is making cards for every teacher, no?
Yeah.
What are the mom? Okay, what's doing it? Video
Sir, the result is on a video again.
how the name?
Nanny.
You can site to get in.
Done as an email like even a die go koelari die.
Okay. Okay,
So
Michelle,
site
why
take a
don't you
mogade?
explain? I don't know. Just check
I'll
with
check.
that message. It's there.
I'll check.
Michelle, can you explain to us about glycolysis in brief, like this? What we learned last week. Can you explain in brief?
Glycolysis is the splitting of glucose into two pyruvates, and ATP is needed for the first steps. And first, the phosphorylation of glucose takes place. Glucose is a 6-carbon compound. And here 2 ATP is used up and it's turned to 2 ADP. And phosphorylated glucose, which is hexose biphosphate, is formed. This phosphate. And then that is split into 2 3-carbon compounds, which is triose phosphate. And thirdly, the oxidation of the triose phosphate forms 2 pyruvate compounds, which is also 3-carbon. And here dehydrogenation takes place, and 2 NAD is converted to 2 NADH. And so along with 2 pyruvates, 4 ATP molecules are formed and 2 NADH.
Yeah. So basically, we are in the lesson of respiration, children. I told you respiration has main 2 types. at aerobic and anaerobic respiration. So main objective is to produce energy. That is why the respiration is conducted by many of the animals. So when it comes to respiration today, so we discuss basically anaerobic respiration as a start, and it has 3 steps. What were the 3 steps? Nishika, did you watch the last week video recording?
Yes, sir.
Can you tell me what's the main 3 types of aerobic respiration?
Glycolysis, Krebs cycle, oxidative electron chain reaction.
Very good. We have glycolysis, Krebs cycle, and oxidative electron chain reaction. So that's the main, those are the main 3 types. And we first moved into glycolysis, which is common pathway for aerobic respiration and anaerobic respiration. Why we call this as a common pathway for aerobic and anaerobic respiration? Rachel, can you tell me why? Why we call this as a common pathway? Aerobic and anaerobic respiration.
It's a common path.
What is the reason? Yeah.
It's because every sort of respiration has to go through those 2 pathways only.
Exactly. Why? Anyone has a different answer? I'm asking why we have to go through that path. How it became common for aerobic and anaerobic. Because aerobic uses oxygen, anaerobic doesn't use oxygen. So how come the first process conducted by both? Rivit, you have answer? Yes, very good. Rivit has stated the answer saying because it can take place without oxygen. That is the main reason. Glycolysis can happen without oxygen, right? That is why. So main glycolysis plan is to convert the 6-carbon glucose into 2 pyruvate molecules. So pyruvate is having how many carbons? 3 carbons, right? Pyruvate having 3 carbons. So glucose 6-carbon molecule break into 2 3-carbon molecules, which is called 2 pyruvates. So it happens in a chain kind of reaction, which we discussed in 3 times, 3 steps. First one, what happened was phosphorylation of glucose. First, the 6-carbon glucose was converted to a phosphorylated glucose where the glucose bound with 2 phosphate groups. So that is what we call phosphorylated glucose. So the glucose name is called hexose bisphosphate. Why we call hexose? Because it having 6-carbon, bisphosphate because it has 2 phosphate groups. Okay. Then in this process, glucose to bind with 2 phosphate groups, it needs 2 ATPs. So we have to waste 2 ATPs in this process. First step. Okay. Now the hexose bisphosphate is produced. Now, second step is splitting the phosphorylated glucose. Now we have to convert this hexose bisphosphate, which having 6 carbon, into 2 3-carbon molecules, which is called triose phosphate. But nothing happened to the phosphate groups. Phosphate groups are there. Bisphosphate converted to what? 2 triose phosphate. 2 triose phosphate. Third step, it's oxidation of splitting glucose to pyruvate. It's just removing those pyruvate molecules, the phosphate molecules from the triose phosphate. So when we remove those phosphate from those 2 triose phosphate, we can produce 2 pyruvates. 2 pyruvates are removed today. They form, right, one triose phosphate produce 2 ATP. So since there's 2 triose phosphate, they produce another 2. So altogether there's 4 ATPs produced. And since they are removing hydrogen in this process, okay, 1 triose phosphate to convert to pyruvate they have to remove hydrogen. So hydrogen, one should take that hydrogen. Someone should take that hydrogen. It is conducted by a reducing agent called NAD. So NAD take that hydrogen to form NADH. So since there's two molecules, two triose phosphate, two NAD each, right? Is NAD will be used to produce two NADH. So altogether, what are the products? The end of the glycolysis, the glucose, 6-carbon glucose converted to what? 2 pyruvates, 4 ATP, and 2 NADH. Okay. What is the net profit of ATP? Even though 4 ATP is produced, what is the net profit? Minadi, can you tell me? What is the net profit?
2 ATP, sir.
2 ATP. Why? At the first step, however, 2 ATP will be used. At the end, there's 4 ATP produced. We remove the waste ATP, then only 2 ATP will be the profits. Okay, it's the profitable ATP amount. So this is what we discuss. Yes, Rachel.
So when what do you call those two triose phosphate break? Do they have like hydrogens free as well? Like do they have like free?
Yes, yes. They have free hydrogen.
Okay, so each
Okay.
time a phosphate group breaks, hydrogen is produced.
Yes. Yeah, exactly.
Okay. Thank you, sir.
Okay, right. So by removal of hydrogen only, they can actually form the pyruvate. So when they remove the hydrogen only, they can form the pyruvate. So that removed hydrogen should be taken by someone. That is where the NAD act as the reducing agent. And there's enzyme also used, dehydrogenase. So dehydrogenase, that means removal of hydrogen. That is what the purpose of dehydrogenase, removal of hydrogen. Okay, that converts the 3-carbon triose to 2 pyruvates. Okay. Okay, so that's the end of glycolysis. Right. Now, this pyruvate, the newly produced pyruvate function, it's not over. Okay, the function is not over. Right. So glycolysis happens where? Happens
In the cytoplasm.
no, the cytoplasm. Right. Glycolysis happens inside the cytoplasm. Now, it has to go to a particular organelle where the cellular respiration happens. What is the organelle? Where the cellular respiration happens? Organelle. What is it, Pute?
Mitochondria.
Mitochondria.
Yes, it should go to the mitochondria. It should go to the mitochondria. So when it comes to the mitochondria, right? Mitochondria is a double membrane bound organelle. Can you remember? Like this. It's a double membrane bound organelle. They have outer membrane. Okay. They have inner membrane. There's a special name for that. And we don't call them cytoplasm. There's another name for this. Okay. This is the outer membrane. What is the inner membrane name? Can you tell me the name? increase stay. Very good. Vivek. Happy. And what is the inner part is called, Michelle?
Matrix.
Matrix. The inner part is called the matrix. So what happened is, here in glycolysis, what are the products that are produced? 4 ATP we have to remove. However, there's 2 ATP. 2 ATP and 2 pyruvate. Because 4 ATP, they have wasted already. So altogether, there's only 2 ATP as a profit. And 2 Na. Those are the products. And ATP, NADH used to Na. So only thing, these 2 pyruvates, right? These 2 pyruvates should enter to the matrix. These 2 pyruvates should enter to the matrix. Okay, so these 2 pyruvates should enter to the matrix. Right? So you can write it down here. Pyruvate is actively transported across the mitochondrial envelope and into the matrix by the transport protein What is the name of the transport protein, Michelle? Can you remember? Can you remember the name of the protein? The name is pyruvate sympose. What is the name of the enzyme? Called pyruvate sympose. So pyruvate sympose is the enzyme. It's a type of a protein. It's kind of a channel protein that uses energy to allow these pyruvate to transfer to the matrix. Nikam pyruvate right? You have to give an entrance fee. What is that entrance fee? You have to spend 2 ATPs. When the pyruvate wants to enter to the matrix, they have to waste 2 ATP. Sorry, 2 ATP, 1 ATP. For 1 pyruvate, we have to waste 1 ATP. Okay. So since there's 2 pyruvates, 2 ATP need to be used. A okay, so You need 1 ATP molecule transfer pyruvate to the matrix. There's 2 pyruvates. How many ATPs are used? 2 ATPs need to be used. Is it clear? So it is being conducted by a transfer protein called pyruvate symptom. And in order to send 1 pyruvate from the cytoplasm towards the matrix, you have to waste 1. Since there's 2 pyruvates are produced, you have to waste how many ATPs? 2. Okay. So now we are moving to the process called link reaction. What is it? The link reaction. Link reaction. Okay. Before the Krebs cycle, there's a process called link reaction. So make a children, you write this down. Okay, this is regarding the movement of pyruvate to the mitochondrial matrix. Okay, this is okay. Is it clear, children? Yeah, sure.
Yes, sir.
Okay. Question done. Minadi, okay.
Done.
Right. Then we are moving to the next part, the link reaction. So this is the reaction between the glycolysis and the Krebs cycle. Okay, that is called the link reaction. It links the glycolysis with Krebs cycle. So the link reaction it links the glycolysis with Krebs cycle. So that is why we call it as a link reaction. So what actually happens in link reaction? In summary, so like pyruvate is formed. Pyruvate is 3-carbon molecule. 3-carbon molecule like this. It is converted to 2-carbon molecule. It is then converted to 2-carbon molecule. So that is happened in link reaction. So 3-carbon molecule, what's the name? Pyruvate. For the 2-carbon molecule, we call it as the acetyl group, acetyl. We call it as what? Acetyl group. So this is what happened in link reaction. So pyruvate converting into 2-carbon acetyl group, that is known as link reaction. Right? So when link reaction is happening, right? When link reaction is happening, right? There will be 2 processes involved. There will be 2 processes. First one, dehydrogenation. Dehydrogenation. Dehydrogenation means what? It's simple to remember. Removal of hydrogen. That is what you meant by dehydrogenation. Removal of hydrogen. So who's doing that? Example. What is the type of dehydrogenase reducing agent we discussed? It was NAD. Okay. Next one, second process. Decarboxylation. Decarboxylation. That is removal of a carboxylic group. Okay. Removal of a carboxylic. So in link reaction, there will be 2 processes happening: dehydrogenation and decarboxylation. Okay. So remember, in link reaction, carboxyl group and hydrogen atom are removed from pyruvate molecule Okay, so in link reaction, what happened? The carboxylic group and hydrogen atom is being removed. Okay, so again, we can draw like this. Three carbon pyruvate. Three carbon pyruvate. Converts to 2 carbon acetyl group. Okay, so in this process, there will be two things happening. The decarboxylation and dehydrogenation. So who's doing the dehydrogenation? It's by the enzyme called pyruvate dehydrogenase. Pyruvate dehydrogenase. Converting this pyruvate into acetyl. So since they are dehydrogenating today, the hydrogen atom will be removed. That atom will be received by the NAD. NAD converts to NADH. And there will be decarboxylation to produce the carbon dioxide molecule. Carboxylic group को remove करके it forms a carbon dioxide molecule. That is how the carboxylic group produced. So at the end of this link reaction, the 3 carbon molecule converts to a 2 carbon. So what happened to the remaining carbon? It's in the carbon dioxide. Okay, so the carboxylic group leaves as carbon dioxide. Okay, so this is what happened in link reaction. Okay, so link reaction is the reaction that links the glycolysis to Krebs cycle. So in glycolysis, pyruvate molecule will be produced. So it can't enter to the matrix as pyruvate, right? It should convert to acetyl group. So acetylization is conducted by 2 processes, dehydrogenization and decarboxylation. Dehydrogenization means removal of hydrogen atom from the pyruvate. Decarboxylation is removal of 1 carbon atom from the pyruvate. So when the carbon atom is removed or carboxylic group is removed, it forms a carbon dioxide molecule. And the remaining, we call it as acetyl group. And when pyruvate removes a hydrogen, that hydrogen will be received by the NAD to form NADH. So however, this entire reaction is catalyzed by the pyruvate dehydrogenase enzyme. Pyruvate dehydrogenase enzyme.
So where do these hydrogen things come from?
It's in the glucose molecule. Like 3 carbon means this pyruvate putih, right? It's a carbohydrate group, no? So in this carbohydrate group, you know, C6H12O6. So C6, 6 carbon. We don't bother about how much hydrogen is produced, putih. How much hydrogens are there. So how we one can a hydrogen okay? The
Okay, sir, understood.
molecule like andot, there are hydrogen in that molecule. So you don't have to know up to that much deep. All you have to know is if one hydrogen is removed, that hydrogen is captured by NAD to form NADH. That's all. Okay.
So can you repeat that?
Again, what you have to remember is all these pyruvates, acetyl group, all the things we discussed before, right, are carbohydrate groups, are organic compounds. So in these organic compounds, it is definitely they have hydrogen. So one hydrogen removed, those hydrogen will be captured by the NAD as reducing agent. So that is what happened.
Okay, sir.
Right. So is it clear, guys, children, up to here? Link reaction? Vinati, clear?
Yes, sir. Understood.
Right. Nishika, clear?
Yes, sir.
How about Rachel?
Yes,
Rachel,
sir. Yes, sir. Yeah.
okay. Then Sarah,
Yes, sir.
Saseka, are you clear? Steve, I hope all of you are okay with it now. Right? So this is Pute, the link reaction. So copy this down. Oh, Tina put cal. You're gonna idamadi. Ilang bitu egane. Anyone drawing? Minadi? Drawing? With are you writing?
I wrote it, sir. Done.
Minadi? Finished writing or still writing?
No, sir. I'm done writing.
Who's writing? Raise your hand. Everyone done writing? Okay. Now, the acetyl group is produced. So acetyl group, it is 2 carbon group. So acetyl is a 2 carbon group. normal. moderator.
Rasa?
Moderator warangan lagi punya kayak kau.
කවුද?
Kau.
කවුරු ආවද?
Ya kayak kau.
ඒක මම උත්තරයක් දෙන්නේ නැහැ. නැත්තම් මාරි නෑනිස පවුනේ.
Right. So what happened in the acetyl group, Pute? It binds with a new enzyme called coenzyme. Coenzyme A. To form acetyl CoA. Acetyl coenzyme A. So finally, this acetyl group combined with a molecule called coenzyme A. In short form, we call it as CoA, coenzyme A. Form okay. Yeah, I'm at the institute. Yeah, sure. Okay. Yeah. I'll be there around till 5 o'clock. Okay, okay. Right. Finally, this acetyl group combines with a molecule called coenzyme A, form acetyl coenzyme A. So this is the end of the link reaction. So as a summary, what happened? Pyruvate converts to acetyl group, and acetyl group converts to what? Acetyl coenzyme A. Okay, so this is conducted by pyruvate dehydrogenase. Pyruvate dehydrogenase. This is conducted by coenzyme A. Coenzyme. Yeah. One NAD. Convert to okay. And also carbon dioxide will be removed. Okay. Is it clear, children?
Sir,
Yeah.
two of these acetyl that thing are formed.
Acetyl coenzyme A.
Ah, yeah, that thing. Yeah.
Not two, only one. From one pyruvate, one acetyl.
One. Okay, okay.
වචන කියාගන්න බැරිද?
Nane, but here they're saying COA to form two.
කොහෙද?
මෙතන පොඩ්ඩයි. මේ 2 එක මොනවාද?
Finally, this acetyl group combines with the molecule called coenzyme A to form to acetyl coenzyme A.
මම හිතුවා form 2 කියලා. TW. ගණන් ගන්න එපා.
Okay, guys, are you with me? Is it clear? Minadi, Nashika, clear?
Yes, sir.
Sarah,
Yes, sir.
Zera, is it clear?
Yes, sir.
අපිටත් කියලා hina වෙන්න ඕන.
ඒක කියන්න බෑ. ඒ හිනාවට ලොකු අර්ථයක් තියෙනවා.
මොනාරි සතෙක් ගැනද?
සුදයිසි.
Okay, okay. It's okay.
These days I'm watching a Turkish TV series.
So මට පේනවා ඔක්කොම ටික.
මොනවද පේන්නේ?
සර් බලන්නේ දැන් මට YouTube බලන්න බැරිලා ඉන්නේ ලැප්ටොප් එකේ.
ඒක තමයි මට ආව Google account එකේ මේනවාද කියලා.
ඒ කියන්නේ?
Someone has used my Google account කියලා මට ආව the notification.
නෑ එහෙම එන්නේ නැහැ. එහෙම එන්නේ නැහැ.
ඔයා log out වුණා නේද?
log out උන්නේ නැහැ සර් නැත්නම් manage කරන්න බෑ ඒක. මම ඉතින් පාවිච්චි කරන්නේ නැහැ ලැප්ටොප් එකෙන් දැන්.
ඔයා කියන්නේ මේ YouTube එක manage කරන්නද?
ඔව් ඒක manage කරන account එකේ එපේ.
හරි.
බය වෙන්න එපා සර් මම මොනවද කරන්නේ. සර් ක්ලාස් එක ඉවර කරන්නේ නැද්ද? අද ක්ලාස් එක ඉවර කරන්නේ නැද්ද?
විසුද්ධව වෙනවලු.
නෑ සර් ක්ලාස් එක පස්සේ මොන හරි වැඩක් තියනවද?
බෑ මම පොඩ්ඩක් කතා කරමු ඔයාලට.
I want to show you something.
Okay, children, shall we move to the next big part, which is known as the Krebs
සර්
cycle?
ඉරේස් කරේ මම අන්තිම වචන දෙකක් තියෙනවා ලියන්න.
Then,
සර් පයිරුවේට් කියන එක හයිඩ්‍රජන් එකක් එළියට අයින් කියන්නේ අයින් වෙලා ඇසෙක්ට ඒ ගෲප් එක හැදිලා අර කෝඒ කියන එක හැදෙනවා.
ඔය එකත් එක බයින්ඩ් වෙලා තමයි ඇසිටයිල් CoA වෙන්නේ.
හරි. Okay.
Right. Then we are moving to the Krebs cycle. Krebs cycle. So write down the Krebs cycle is the next stage of aerobic respiration. Aerobic respiration. And it takes place in the mitochondrial matrix. Happens in the mitochondrial matrix. Okay. In the Krebs cycle, a series of enzyme catalyzed reactions occur to oxidize the acetyl coenzyme A. See, CoA කියලා මතක තියාගන්න it's acetyl coenzyme A. Produced in link reaction. Okay. So next point. In Krebs cycle, ATP molecules are wasted and coenzymes are used as reducing agent. What are the types of coenzymes that you know already? Now you know first coenzyme, that is NAD. Then here at the link reaction, I told you there's a coenzyme A. That's another one. Now here there will be a new one. We'll be learning. Example. Example. NAD and FAD. NAD and what? FAD. Okay. Also, right? What is status them? Hot. Idine balla. Also, carbon dioxide will be formed. Will be formed. Okay. So, however, as a summary, you remember one glucose molecule, it breaks into how many pyruvates? 2 pyruvates, right? 2 pyruvates. One glucose molecule breaks into 2 pyruvates. So each of these pyruvate converts into what? Acetyl coenzyme. Okay. The link reaction. Acetyl coenzyme. So for one glucose, how many link reactions should happen? For one glucose molecule? Huh? Tell me. One glucose molecule, how many link reactions should happen? Huh? David, for one glucose molecule?
2.
Yes, very good. There should be 2 times link reaction. Then tell me how many Krebs cycles should happen? Krebs cycles also happen. Krebs cycle like හැදෙන products ඔක්කොම double වෙනවා when you want to find the total outcome, okay, from one glucose. So keep that in mind always.
So ඒ කියන්නේ මොකද්ද?
ඒ කියන්නේ දැන් අපි හැම වෙලාවෙම මෙතන කතා කරේ එක glucose එක pyruvate දෙකකට කැඩුනට we went through from one pyruvate converts to acetyl, that acetyl converts to acetyl CoA පුතේ. But you have to remember one pyruvate is not glucose. Glucose is composed of 2 pyruvates. That you have to keep in your mind. තේරුණා?
Yes.
Like a glucose molecule, one glucose molecule has to go how many times to complete the entire breakdown?
4.
Eh? One glucose molecule, how many link reactions it should go?
2.
2 link reactions. How many Krebs cycles it should go?
2.
So one link reaction produces one product, and one Krebs cycle produces one product. So if there's 2 reactions, all the products also become double, no? Keep that in mind. Is it clear?
So two carbon dioxide molecules will be formed.
Oh, ඒක තමයි මං කියන්නේ.
හරි.
Okay. Right. Now, let's start with Krebs cycle. So,
No,
Krebs
aren't we going to
cycle.
write that?
Sorry?
Not going to write the previous. Yeah.
You are?
නෑ ලියන ගමන්.
පොඩ්ඩක් වෙලා වැඩියෙන් ඇයියා කට්ටිය. Because I don't want to stop this in middle. එහෙන දේශේ එන ලෝක දවසත් තමයි කොරෝනා නැති දවස්.
Done, sir.
Okay, others done? Okay. That you have given the correct answer. Now only I saw that. Right. Now this Krebs cycle, it's called a cycle because it has a cyclic nature. Okay. So you have your A-level note, right? So in your A-level note, you can see okay. The Krebs cycle. Okay. So when it comes to the Krebs cycle, Pute, what? Yeah, මේක තමයි Krebs cycle. මේක ඔයගලන්ට පාඩම් වෙන්න. This is what we discussed. Link reaction. The pyruvate converts to acetyl-CoA. There, NAD will be used as a reducing agent to form NADH, and carbon dioxide molecule will be used, and 2-carbon acetyl group will be produced. Okay. As stepwise, we are going to learn this. The main balapam pissu hadana thama. Neda
Sir, please ඇදලා ඉවරයි sir.
කලබල වෙන්න බබ stepwise yeah. Right. So stage 1. Stage 1. The acetyl coenzyme A will break down to form acetyl group. And what? Coenzyme may separate. ඔක දැන් කඩනවා දෙකට. අර අන්තිම එකේ සෙට් වුණානේ. link reaction එකේ. දැන් ඔක ආයේ දෙකට කඩනවා. Okay. So they have the coenzyme, acetyl coenzyme main. Coenzyme main. It will be converted to what? acetyl group plus coenzyme A. That is the first stage. That's not difficult. No? Difficult than stage 2. Stage 2. The 2 carbon acetyl group then combines with 4 carbon compound called oxaloacetate to form 6 carbon compound. 6 carbon compound called citrate. Now here we have broken the acetyl group, the acetyl coenzyme A back to the acetyl group. Now what we do is stage 2, this 2 carbon acetyl group will be bounded with 4 carbon molecule called oxaloacetate. This is acetyl group. What is this? 4 carbon. Oxaloacetate. They forms what? They form 6 carbon compound called citrate. So, citrate. This is 2 carbon, 2C. This is 4C. Citrate is how many C's? 6. 6. Okay. So first stage, acetyl coenzyme will be the coenzyme will be removed from the acetyl group, and second stage, acetyl group will bond with 4 carbon oxaloacetate to form a 6 carbon group called citrate. Okay. Okay, done. Vivit, done.
Not yet, sir.
Are you disappointed, Vivit, on something?
No.
Okay.
No, sir. Why?
Okay.
Yeah, I'm okay.
But
No, I'm fine. I'm all right.
okay, children, shall we move to the next level? Okay, Vivit.
All right, sir.
Stage 3. Stage 3. So now what has been produced in stage 2? Citrate. 6 carbon citrate. Will be decarboxylated and dehydrogenated. Decarboxylated and dehydrogenated to form a 5 carbon compound. Just have to remember 5 carbon compound. Plus, now if it has decarboxylated, definitely they have to produce what? Carbon dioxide molecule, right? The carbon dioxide molecule. Then if it has become dehydrogenated, there should be what? The reducing agent. Here the reducing agent is NAD. So NADH will be produced in stage 3. So dehydrogenated, who's doing it? මගේ status එක දාගෙන මේ කට් එක දානවා නේ respect එකට මොකද self respect අපත් කවුරුත් කිව්වා කොහොමයි right. So citrate 6 carbon. In stage 3, the citrate will be decarboxylated as well as dehydrogenated. So if they are dehydrogenating, definitely there should be a reducing agent. So here the reducing agent is what? NAD. So NAD converts to NADH and decarboxylated produce a 5 carbon compound and carbon dioxide and NAD. So this is what happened in stage 3. Okay. Now, stage 4. Stage 4. Stage 4. This 5-carbon compound is then further decarboxylated and dehydrogenated. I para karavede is then further decarboxylated and dehydrogenated. So now there is how many carbon? 5 carbon. 5-carbon compound converted to what? Converted to 4-carbon compound. And if it's getting decarboxylated, definitely the carbon dioxide should produce. Am I right? And here also the reducing agent is what? NADH. ආස නැති පාට් තියලා බලන්න ඉච්චර. ඔය බලන්න නැත්තම් බලන්න නැත්තර මොකද හේතුවක් තියෙනද ඔයා සමහර උතුත වෙඩිකමට බලන්නේ. සමහර උසු. මේකද? ෆෙන්ස් එක. ඔව්. ඒක තමයි Joey Chandler, revenue apartment, Joey, Phoebe വെച്ചതാ. Yeah. Chandler people. Phoebe Chandler. Chandler. Okay, done, children. With Ranavir, okay?
What, sir? Yes, it's all
Right.
right.
Sarah, are you with me? All good?
Yes, sir.
Okay. Next one. Stage 5. Now, what is the number of carbons? Of 4. So with here, this 4 carbon compound, then temporarily, temporarily, temporally, temporarily combines with O enzyme A. And in this stage, ATP is produced. So our O enzyme may carbon bending hinda, they will produce ATP in this stage. Okay. So we have carbon compound. Yeah. So coenzyme. This coenzyme E. They both and then joins together. But this product process, ATP is generated. ATP is generated. Yeah, so right, a special point here. Okay. So here the ATP produced because of the 4 carbon, right? So this is a new term, right? This production of ATP in Krebs cycle is called substrate level phosphorylation. So what is the substrate here? The 4 carbon compound. So they remove the phosphate groups to bind with ADP once it reacts with CoA. So that ADP can react with the phosphate to form ATP. So that's why we call it as substrate level phosphorylation. Okay.
Anessa, can you explain that part again?
You don't have to know much about that actually, right? All you have to know in stage 4, it is the substrate level phosphorylation. But when you go to the next cycle, right, that is the oxidative electron chain reaction, you will learn a new phosphorylation type called oxidative phosphorylation. එතකන් දැනට මතක තියාගන්න, in stage 5 of Krebs cycle, the 4 carbon compound, when it reacts with the coenzyme A, okay, it releases a phosphate group. Okay, so that phosphate group binds with the ADP to form ATP, right? So this reaction, it forms, it generates energy. So that's why we call a substrate level phosphorylation. Okay, stage 5, the 4 carbon group binds with the coenzyme A. So there, the phosphate group is removed. That phosphate group is captured by ADP to react to form ATP. And this reaction is called substrate level phosphorylation. Is it clear, Vivit?
Yes, sir.
Okay. Then stage 6. Stage 6.
Sir, stages කීයක් විතර තියෙනවද?
ඉස්සරේ දාන්න. A level tutor දෙන්න. ඔය විසංකගේ A level Cambridge නම දාලා තියා. පොඩ්ඩයි නම. Seven one 817 537. Wait. Okay, Bhute. So we are moving to stage 6. So in stage 6, the 4-carbon compound. Let's see what happened to the 4-carbon compound. So since it's a temporary binding of coenzyme A, Bhute, once the ATP is produced, the coenzyme will be removed, right? So they got point to that. Okay. Once the coenzyme, once the ATP is generated, coenzyme A will be removed. Okay. Now it comes to stage 6. Stage 6, the 4-carbon. The 4-carbon compound is dehydrogenated. 4-carbon compound is what? Dehydrogenated. Okay. So there's the 4-carbon compound. Okay. It's the 4-carbon compound. Carbon compound will be dehydrogenated. So if there's dehydrogenation, then definitely you know there should be a reducing agent, right? Okay. Now there's a new reducing agent. I taught you in the beginning. There's a new one here. What is the new one? FAD.
FAD.
So here, FAD converts to FADH. So from the dehydrogenation, nothing will happen to the carbon number. carbon number remains the same, but they will produce the FADH. You learn this like for your higher studies, right? Medicine වගේ යද්දි මේවල හැම එකම නම් තියෙනවා. ඔයගලන්ට ඉතින් ඉච්චර නැහැ. ඔයගලන්ට 4 carbon compound, 3 carbon compound කියලා තමයි තියෙන්නේ. හරිද? Real glycolysis Krebs cycle එක ගහලා බලන්න in internet, you will see each one of these 4 carbon, 3 carbon, 5 carbon has names. So you will have to know those things. මනත් මතක නේද? Right? Right? So in stage 6, what happened? 4 carbon compound is dehydrogenated. Here the reducing agent is what? FAD. So FAD removes a hydrogen to form FADH. So 4 carbon remains the same. That is stage 6. Okay. Shall we move to the stage 7? Which is the final stage. We should be happy now.
Yeah, sir.
You really thought that there's 10 stages, no?
Yeah.
Okay, shall we move to the next one? Anyone writing?
The classes but the breaks even they net the bit.
The break part of the myself like you will.
So Gia the go Harry.
Manusakal use the link.
I have that.
I will have a
Nah, man, yeah.
Okay, but shall we move on to the next one? Shall we? Okay. Stage 7. Stage 7. The new 4 carbon compound is then further dehydrogenated to produce oxaloacetate. Further dehydrogenated, produce what? Oxaloacetate. So metana reducing agent අපි පරණ NADH ම තමයි. But there's an enzyme which does this. Enzyme called isomerase. Isomerase. Enzyme isomerase. Nice. The reaction. Okay. So both are 4 carbon. This is also 4 carbon, but we call it as a name as oxaloacetate. Oxaloacetate. Okay, so there'll be a product. Right. Now, in summary, tell me, the cycle can then continue again. Why it should continue again, Pute? Because we did this for only one pyruvate molecule, remember? However, for glucose, there's 2 pyruvate molecules. So it should continue once again, right? Okay. This cycle can continue again since it has 2 pyruvate molecules generated from one glucose molecule. Clear? So, however, for one pyruvate molecule, what are the products in Krebs cycle? How many NADH are there? Someone tell me. How many NADH are produced? Tell me. 6? No, for one pyruvate. For one pyruvate?
3.
3. How many carbon dioxides? 2. How many ATP? How many FADH? Okay. So for one, this is for one. One pyruvate. One pyruvate. Is it clear, children? මේක ලියාගත්තද?
Yes, sir.
Finish writing?
Yeah. තාම මොනවත් නැහනේ.
තව තියෙන එක දෙයක්.
Okay.
Others finish writing? Michelle?
Yes, sir.
Minadi? Done?
Yes, sir.
Okay. So we go from the top. Stage 1. What happened? Now from the link reaction, they produce what? Remove the coenzyme A part to form acetyl group. Okay. In stage 2, what did they do? The acetyl group was joined with oxaloacetate, which is a 4-carbon group. They form a citrate, which is 6 carbon. That is stage 2. Then stage 3, citrate 6 carbon will be decarboxylated, dehydrogenated. Decarboxylated produces NADH. So in the stage 3 and stage 4, altogether there's 2 carbon dioxide produced here. You can see. Then stage
Excuse me,
5.
sir.
Yeah, sure. Tell.
Doesn't acetyl coenzyme A and oxaloacetate combine to form citrate
In which reaction?
in Krebs cycle, like right after link reaction? Doesn't acetyl coenzyme A combine with oxaloacetate to form citrate?
No, not the acetyl coenzyme. Acetyl group reacts with oxaloacetate.
Okay.
එම ඒක නම අපි ඉස්සලම් පටන් ගත්තේ. Acetyl coenzyme එකෙන් තමයි matrix එකට එන්නේ. ඊට පස්සේ coenzyme මේ කැලල ගලවනවා. That is the first stage. Then the acetyl group binds with the oxaloacetate to form a citrate. My keyword is at the beginning.
Yes, sir.
Okay. Right. Then stage 3, you heard what I said. 6 carbon converted to 5 carbon and carbon dioxide and NADH produced. And then stage 4, 5 carbon compound further decarboxylated and dehydrogenated, forming 4 carbon compound and carbon dioxide and NADH. Now it has turned to 4 carbon. Stage 5, the 4 carbon here, temporary binding of what? Coenzyme A. Temporary binding of coenzyme A produces a ATP. So that is called substrate phosphorylation. Stage 6, what happened? The 4 carbon compound further get dehydrogenated. Here the new reducing agent was used, FADH. And 4 carbon and FADH is produced. Stage 7, finally, the 4 carbon further get dehydrogenated by NADH, and there's an enzyme that catalyzes this action, right, to form oxaloacetate and NADH. So altogether, the products from one pyruvate molecule, they produce 3 NADH, 2 CO2, 1 ATP, and 1 FADH. Okay, right. Then, api balamu. Now here there is what? Acetyl coenzyme A. Now acetyl coenzyme A, what is the first step? Acetyl coenzyme A breaks into what? Acetyl group plus coenzyme A. Understood? Right. Then the acetyl group binds with oxaloacetate to form a 6 carbon citrate. 6 carbon. There are 2 carbon. Okay. Six carbon. Then they make a citrate powder pathway. Then what happens to the citrate? It get decarboxylated. Either and dehydrogenated. Dehydrogenated. What is the reducing agent here? Tell me, children. There will be 2 products, carbon dioxide and NADH. Either. Right. Then when they form what? 5 carbon compound. Okay, 5 carbon compound. Further decarboxylated and dehydrogenated, produce what? 4 carbon compound. මෙතන මොනවද දෙන්නේ? Carbon dioxide and NAD. Okay. Then what happened to the 4 carbon compound? It converts into again 4 carbon compound, but dehydrogenation happens. Okay, but the process is called dehydrogenation. New one, new reducing agent FADH. Okay, then the 4 carbon compound further converts to 4 carbon compound. Carbon compound called oxaloacetate. Carbon compound oxaloacetate. Here, what is generated? What is generated? NAD. Then පටන් ගනිද්දි මම මොකද්ද කිව්වේ? Acetyl group reacts with oxaloacetate කියලා කිව්වා නේද? බලන්න ඒක නෙමෙයිද මේ මෙතන වෙන්නේ කියලා. Oxaloacetate reacts with acetyl group again. Here, citrate. This is the Krebs cycle. Okay, උච්චරයි තියෙන්නේ මහ ලොකු මේ මගුලක් නෑ. All you have
So
to
what
do.
about the release of ADP?
I forgot. ATP is released where?
Stage 5.
Stage 5. Step up another.
Sorry, FADH එකට ඉස්සෙල්ලා.
FADH එකට ඉස්සෙල්ලා ඕනේ. Second. Second. So what I'm saying is 4 carbon compound. Carbon compound that will react with what? React with coenzyme A. And during that reaction, they generate AT. මොකද වෙන්නේ? 4 carbon compound. Converts to further 4 carbon. Sorry, Matega. Thank you, Ravit, for reminding me.
No problem, sir.
Right. So this is the Krebs cycle. So if you look like this, it's terrible. I know it's difficult to remember. But if you go through the stepwise, then you should be able to plot it in a table like this. Okay. So you have to study this. You have to buy hard. It's not easy, I would say, but it's not difficult. Right? Just you have to remember the steps. Okay. You have 2 cycles. You have to remember the glycolysis, the the link reaction, then the today discussed Krebs cycle. Okay. Right. So children, talk to me. Is it clear what we discussed for today?
Yes, sir.
Yeah.
Yes, sir.
Yes.
Okay. So study well. Hope to see you guys next week then. Okay. God bless you. Thank you.
God bless you, sir. Thank
Thank
you,
you, sir. God bless you.
Thank you, sir. God bless.
God bless you.
Thank you, sir. God bless.
Sir,
Yeah.
hi.
What?
The pain sir. අපි අර මේ මොකක්ද අර වීඩියෝ එකට අපි මොනවද කරන්නේ? තව එකක් දාන්න ඕනේ නේ.
මේ වීඩියෝ එක නෙමෙයි දැන් ප්‍රොමෝෂනල් වීඩියෝ එක පොඩ්ඩක් ප්ලෑන් කරමු.
හරි සෙට් කරමු. කොහොමද හදන්නේ මේ